Hello everyone, my name is Abigail Desch and I will be presenting on behalf of the 2021 to 2022 PHAGES program here at Drexel University. So I'm sure we're all familiar with kind of the layout of the PHAGES program. Some things that were unique to our experience here at Drexel is that we were able to isolate 42 novel bacteriophages on Microbacterium Foliorum. And from those 42 phages, we were able to sequence the DNA of 5 phages, all of which were lytic. And then in order to ensure that our work in winter term was accurate with all of the start site and gene function annotations, we employed our phage cup competition. And I'll go into greater detail of kind of how it functions later in the presentation. And then finally, I'm going to talk about our future plans and the research topics we will be working on this final term here at Drexel. In our fall term, we went through isolation, purification, and extraction with our bacteriophages. In the middle, you can see an example of one of our serial dilutions. We had some hiccups along the way when we were doing serial dilutions. There was some mix-ups with bacteria, so there would be times that we would have to go back and repeat serial dilutions because of the great host specificity that comes along with bacteriophages. Like I said before, we ended up getting 5 phages that had a high enough DNA concentration, and we then had them sequenced at the University of Pittsburgh Bacteriophage Institute. And we would like to thank them greatly for all of their help throughout this year. Then in the winter term, we then did bioinformatics. They were called Fireproof, Fred Flintstone, Crazy Rich, Forgius, and Binkbonk. 2 of these were in the EA1 cluster, 2 were EC, and 1 was EE. Basically, how we did our bioinformatics was that each section had one of the bacteriophages to do the start site and gene function annotations for, and then Binkbonk, the final bacteriophage, was given to all 4 sections and annotated by all 4 of them and then compared within. Results that were found from our bioinformatics was that in Binkbonk, there was actually a frameshift mutation in gene 13. We also thought that gene 26 in Binkbonk might have coded for tRNA. It was ultimately found that it didn't. It likely codes for the same protein as gene 25. So as I mentioned, Binkbonk was given to all 4 sections, and then we were able to compare our annotations. And this was called the phage cup. So these annotations were really conducted independently within the sections. We were left to kind of self-organize and figure out how we were going to divide the annotations between everyone in the class and how we were going to peer review within our class before eventually the annotations from our class would be sent to a different section for them to correct. So we could earn points for getting correct annotations, but we could also earn points for showing evidence of team building. The example that you see on this slide is that one of the sections dressed in all black. And then once we received the annotations from a different section, we were able to take away points from that section if they had incorrect annotations. So throughout the entire year, we have all had an incredible time in PHAGES. We are forever grateful that we had this opportunity to conduct such interesting and novel research in our first year of college. And this in our fall term, when we had different difficulties with the bacteria or when we had to go virtual because of the pandemic, and we learned how to overcome and work together. And we hope that we can continue this spirit in our spring term when we will be working on our research projects. And these research questions will be conducted on the bacteriophages that we isolated throughout the year. And we had to submit a project proposal before spring break that detailed our research question, some background information, what our experimental procedures would be, and then the importance of the research. And we're all greatly looking forward to see how our research turns out and also what the other people in our sections are doing, since we have built such a sense of community throughout the year. And then finally, I just want to thank everyone who has made this year possible. Really importantly, all of the professors, the prep staff, TAs, and students at Drexel University. I thank the PHAGES team, the Howard Hughes Medical Institute for making this program possible, and Drexel University for offering this program to us in the first place. And of course, I want to thank all of you guys for listening. Thank you.